Okay, so when we're talking about DNA synthesis, we're taking a DNA strand, DNA polymerase binds to it in order to make another DNA strand. When we're talking about RNA synthesis, that's the same thing as transcription. So we're taking a DNA strand, opening it up, an RNA polymerase comes in, adds in the corresponding nucleotides, and in the end, we're going to get an RNA molecule. So essentially, we're taking something and making something else out of it. So we're taking a DNA strand to make RNA, which will be used for further processing in translation. Okay, so when we're talking about translation, we are taking, this is when the mRNA, which was synthesized in transcription, leaves the nucleus, enters the cytoplasm, and finds the ribosomes that are throughout. When the mRNA finds the ribosome, it binds to the smaller subunit of the ribosome. The ribosome has a small subunit, a larger subunit, and different rRNAs or ribosomal RNAs throughout it. We'll talk about those later. The mRNA, when it comes, it kind of just waits there until something else comes. Now, in other processes, we've had DNA polymerase, RNA polymerase, but this is in translation. What is crucial is the tRNA, which is throughout the cytoplasm, and when it comes and it binds to a specific substrate, which is the amino acyl tRNA synthesis. And that's when a tRNA gets its appropriate amino acid to the end of it. And that will move into the ribosome. And that's kind of like being, the main thing that needs to kind of enter the ribosome, because that will bring the amino acids in, and then they could form the polypeptide chain and lead to the proteins. So after the tRNA has been activated by this specific amino acyl tRNA synthase, it has it's kind of like this, and it has amino acid on top, and on the bottom there are 3 nucleotides. Those are the anticodons. Those are they're very important, because when the tRNA comes into contact with the mRNA and the units of the ribosome, those anticodons will match up with the codons on the mRNA. The mRNA codons are 5' to 3', and so that means the anticodons have to be 3' to 5'. One codon codes for one amino acid. One amino acid has many different codons that code for it. It's kind of like the anticodons just want to be included. So they come, they match up with their codons and say, hey, I want to join the party. Let's get in on this situation. And that situation is protein synthesis. Okay, so we have the small unit of the ribosome that we were talking about, the mRNA, which is with that, and then we have the tRNA coming in, and now the codons are matched up. Now, what comes next is the larger ribosomal subunit. The tRNA, the mRNA, and the ribosomal subunits are the translation initiation complex, and that kind of initiates, those are the ingredients for the rest of translation to occur. You have the information you need. Now all you just have to do is put it together. Consider the ribosomal subunits, small and large, mRNA and tRNA, as like the lights, camera, action of translation and protein synthesis. Okay, so when the large subunit of the ribosome comes, it has 3 different kind of like alleyways. In the middle, you have the P site, and then you have on the 2 ends an A site and an E site. The P site holds the tRNA with the amino acid and the codons on the bottom, anticodons on the bottom. So once the tRNA comes and binds to the A site and the polypeptides kind of join in, it's able to transfer itself, translocate down the strand of the mRNA. Then once it moves, it's the one in the P moves down, and then it exits through the E site when it's not really needed anymore. But once and then it reaches a stop codon, and that's when everything stops. But what's really important is as each tRNA is coming in, it has its own amino acid that with its anticodons, and there's a polypeptide bond that's forming because they're being transferred. Now, one's being held in one alleyway, one tRNA molecule is being held, then the other one is moving down and is able to transfer its amino acids to the other on the strand, and they're able to make a polypeptide chain. So at the third step of translation, so it was the initiation when everything came together, the elongation, which is kind of polypeptides coming all together, moving down. Now the third and the final step is the termination, when the tRNA reaches and terminating an end codon, a stop codon, it breaks away and is able to kind of move and do its thing in other locations throughout the cell. That's at the stop codon after it leaves through the E site. And this whole process is carried out by GTP. It was kind of like ATP in like cellular respiration, but this is the GTP. The endergonic reaction, through it, it's able to lend its energy and that's needed through the process. I really hope that this is how you can translate translation. And on a side note, I have written the stop codons on my hand. That's when you know you're a pre-med student.